Hello, I'm Jasmine Godbold. I'm Professor in Marine Ecology at the University of Southampton. And I'm a new member of the Scientific Advisory Group for INSITE 3. And I'm going to be chairing this session. So to kick us off, we've got Debbie Russell from the Sea Mammal Research Unit. And she's going to talk to us about the INSITE's Phase 2 project, EcoSTAR project updates.
Thanks very much. Thanks for the introduction. So I'm going to talk a little bit about EcoSTAR, which was one of the INSITE 2 projects. I just want to say it was a big collaboration, especially between SMRU at the University of St Andrews and CFAS, with lots of partners and also collaborators. I want to especially acknowledge my co-author here, Matt Carter, who is a postdoc who led a lot of this research but couldn't be here today. So in terms of a project overview altogether, essentially this project came out of INSITE Phase 1. So there was a few of us that were involved in INSITE Phase 1, and we came up with these outstanding key knowledge gaps that we brought together into this EcoSTAR project. The key thing in terms of, I'm not going to talk about today, is the benthic functioning and also the ecosystem modelling. So that's using EWE that you've heard about a bit today to look at North Sea-wide ecosystem model. Those were updated on last year at this conference, and so I'm not going to focus on them, but instead focus on the marine mammal aspect. So these guys here. And in the interest of time, I'm going to focus even more down into distributions and foraging behaviours, and particularly just on seals. Okay, so first question really, kind of the basis was just where the structures were, which was a little bit more tricky than we had hoped in terms of getting information on the where, what, when, that we really needed to understand the potential impacts of these structures, especially given the North Sea, whole North Sea focus of our work. And so as a result of this, we came up with a work package 0, which was focused on generating the required data sets with the required metadata that we know can mediate impacts. And this has actually been updated last year. This was work led by PhD student under the super funding. And we're still looking for, if anyone wants to host this, that would be great. So we then thought we would move on into kind of what are the impacts of structures, but actually we also not only need to know where they are now, but where are they going to be in the future? Because obviously we can't predict, with the potential magnitude of these effects without knowing that. And so this was work that was led by Simon Waldman at Heriot-Watt University that I found out about, and essentially through the EcoSTAR funding we got this work finalised. And the point here is not to come up with these are where we think the wind farms are going to be in the future. The point here is to come up with a realistic, feasible landscape, right? So it's not going to be correct, but is on which that we can base our predictions to understand the potential magnitude. So it's using the current and future areas and country-specific targets and wind farm designs. That's available to download at the moment. Okay, so moving on to structures. They're not heard too much about marine mammals, so why do they matter? Well, there's the potential for structures to impact foraging success of marine mammals in various different mechanisms, but also they're seen as indicator species for the health of the marine ecosystem. Importantly here, when it comes to structures, I think we all really need to remember the potential top-down impacts that marine mammals can exert. The 3 most abundant marine mammals in the North Sea, very roughly, and I made this up, back of envelope type stuff, 850,000 tonnes of fish a year, but also that is obviously not spatially, across the North Sea, that's very particular areas. So it's really critical to include them in ecosystem models and to understand their potential impacts at structures. Because in areas of high overlap, if you've got a lot of marine mammals, you've potentially got an ecological trap for fish. You potentially may not find an effective structure because the marine mammals are going there first. So there's lots of aspects, and so we really need to understand the spatial distribution of marine mammals and the impacts of structures on their foraging. And this is critical for understanding and predicting the impact of structure presence and removal, not only for marine mammals, but in terms of the ecosystem as a whole. Okay, so firstly, to do that then, we were looking at estimating the at-sea distribution of seals. And unfortunately, at-sea surveys data doesn't really cut it for seals. And so we use these tags that we glue onto the back of the neck of seals on the fur, and they fall off after the annual molt. And we can integrate this tracking data with information on the environment, so distance from haul out, essentially proxies for prey availability. And we need to do that because, unfortunately for us, not all haul-outs are visited by tagged animals. So we need to come up with this habitat association model. And we can do that in a regional way that then allows us to take the haul-out counts, so we know where animals haul out, and predict the at-sea density, so a snapshot of the distribution. So we did this a few years ago now, partly as part of EcoSTAR, and came up with these distributions for grey seals and harbour seals. However, the issue is that this was only for UK and Irish haul-outs. And so if we want to understand the top-down impact on a North Sea basis, we need to go further. So we need area-wide estimates. And this is also very important for understanding transboundary impacts of various anthropogenic activities. So we were incredibly lucky to get data for all around the continental Europe, in terms of, for the North Sea, get a much better impression of where animals go. So this is grey seals and harbour seals, and the circles show all the tagging information. And this was only possible because of collaborations from every area here. Okay, so our aim then was to predict for the Northwest European shelf, and essentially we incorporated these additional regions I mentioned. We also updated the haul-out counts and included updated tracking data for UK and Ireland, and made some improvements to the methodology to come up with these updated maps for the Northwest European shelf. So essentially what these are in 5-kilometre grid cells, the predicted distribution of seals. So if you drained the sea very, very quickly, what would be the mean number of seals that you would expect in each 5-kilometre cell? So now we know where we think the animals are, we need to understand, well, what does that mean about structures? So we can take that and we can look at what is the percentage of the North Sea total that we would be expecting to overlap with oil and gas. Now here, by we say overlap, we mean within the same 5-kilometre cell. So you can see for grey seals, it's up at above 4%, and harbour seals, it's a bit lower. We can then basically make the kind of catastrophic assumption that the seal distribution has stayed constant through time, which it has not, but just to illustrate, this is all very preliminary work, by the way, that you can then look on how you would have expected the at-sea overlap with structures to have changed through time. So that's for oil and gas, and then you can do the same for wind farms. So you can see this rapid increase with wind farm development, and then you can use the overlap, kind of these feasible overlap levels from the future, from Waldman et al., to then predict what kind of levels of overlap we would expect by 2050. So you can see it's quite high for grey seals in particular because they're more offshore. Okay, but what about foraging? So in terms of foraging, we again use the same tag data, and we can actually use that tag data to infer whether they're most likely to be foraging, so area-restricted search, or transiting, based on their movements. So we can do just an illustration for a grey seal on the left here. You can see the haul-outs in black. The blue is predominantly transiting, and then the yellow and orange is what we'd expect to be associated with foraging, and the same for harbour seals here. Overall, we know that this is most likely driven by natural kind of prey proxies. So at population level, it was driven by potential energy anomaly. I should say driven by, associated with, because these are really proxies. So this is to do with mixing levels, essentially the hilliness of the sea and the substrate. So once we've accounted for the kind of natural environment, we can then think again about structures. So what does this mean for structures? So we have some individuals, this is a grey seal, that clearly associate with structures. So these are oil and gas platforms. And you can see that the probability of foraging here is higher near a structure. It's basically 1. So if you encounter a structure, so you're within 5-kilometre cell, what is your probability that you're actually foraging rather than just transiting? So you can see it's much higher for this individual when they encounter oil and gas. And this is another example for a harbour seal, where again, this is a wind farm, you've got a much higher probability of foraging, given the environment, if you encounter a structure. But when you look at that in a population level, that doesn't really continue. So here on the left-hand side, you've got no structure, and what's the probability of foraging versus travelling? Blue is grey seals, red is harbour seals. And that actually decreases a little bit with structure. And that's to do with this kind of travelling aspect, where it's not that individuals are encountering a structure and then switching from foraging to travelling. It's simply that these areas are often inshore, near what I would call like a highway here. So you're going to kind of, if this blue was in here, you get kind of a more associated with transiting. So we can now combine these with the overall overlap to then project, well, how many animals do we think are going to be foraging in the vicinity of structures now, and how many in the future? So, for example, for grey seals, at the moment, we would expect about 0.5% are foraging within wind farms at the moment, and that would go up to about 1.5% with the projected change 2050. Okay, I don't know how my time's doing. Okay, so I'm just going to conclude then. Sorry, this was quite rushed, but just a few considerations and future research priorities. So EcoSTAR finished in March this year, and we'll be publishing these kind of results in the next little while. But essentially what I do want to say is about these density maps. Essentially, if we're predicting population level impacts of anthropogenic activities, we just need to be really careful when we're interpreting these maps. So these maps are basically a snapshot. They are a mean over seasons, over time of day, over tide. So in fact, if you were at high tide, you could multiply the numbers by about 1.2 because more animals are actually in the sea. So if you have something like a UXO clearance, then actually you can say, well, a mean level of animals that may be impacted by this UXO clearance, so within a range. However, when you have repeated activities like pile driving, you need to understand the turnover of seals. So in these 5-kilometre cells, you might estimate there's 5 seals, but an hour later it could be a different 5 if it's transiting. Whereas if it's a foraging area, it might be some seals actually stay there for over a week. So this is kind of a real issue in terms of the misuse of these maps. What does foraging structures mean for seals and fish? Well, I think that's still open. We've got a PhD student at the moment who's looking at the foraging at structures, and it appears that although some animals do clearly specialise and go to structures, they do forage elsewhere. So it doesn't look like if you remove the structures, they wouldn't have anywhere to go. But structures also result in a change of prey composition. It may be an increase, but it's a change, okay? And change has impacts, whether good or bad. And a PhD student here at the moment, Philippa Wright, is looking into the calorific change. So what happens? Are you getting kind of good quality, lower quality fish? Because obviously sand eels is a key here, which is not really normally associated with structures. So there's ramifications of prey quality, potential top-down impacts on fish, but there's also impact on the competitive landscape potentially if you've got these fish in more kind of limited areas. And just another plea to integrate these top predators in ecosystem models. So we've done this for EcoSTAR, and the results are coming out on that soon. Just want to say thank you to the funders, a huge number of collaborators, and I'll just leave you with this, which is basically a new R-Shiny that people can look at to get interactive estimates for seals hauling out in Scotland only at the moment. Thank you.